Aisyah Nabila binti Muhammad Safri, BFT 240900001.
Hi, my name is Nur Atika binti Kamrin, BFT 240900002.
Hi, my name is Misha Loyapik, BFT 240900003.
Hi, my name is Kristen Chong Siu Yet, BFT 24090009.
Hey, did you just finish class?
Yeah, I'm hungry.
Me too. What do you feel like eating?
Kimchi?
Kimchi? I love kimchi. I like how we don't even have to cook it.
Really? How?
Because it is called fermentation process.
What's that?
Fermentation process is when good microbes like lactic acid turns sugars in veggie into acid.
Sounds fancy. So, does it taste tangy?
Exactly, because the acid helps us to make the food like for safer consumption. It stops the bad microbes from growing. Here, let's go to the lab and I will show you more. So, what is kimchi? Kimchi is a traditional Korean fermented vegetable product. In a microbiology perspective, kimchi is an excellent example of natural lactic acid fermentation, where the microorganisms are already present on the raw ingredient, drive the transformation of the food into a safe, acidic, and shelf-stable product.
Before we start, let me introduce you the ingredients that we need to make kimchi. First, we have glutinous rice flour, we have fish sauce, gochujang paste, onions, garlic, ginger, salt, apple, cabbage, and lastly a clean container.
The first step is raw material selection and preparation. Fresh Napa cabbage is being selected and inspected to ensure it is free from physical damage and spoilage. Using fresh vegetables reduces the initial microbial load and limits the presence of spoilage microorganisms, creating better conditions for controlled fermentation. Next, primary washing. The cabbage is washed thoroughly under running potable water to remove soil, dust, and surface microorganisms. This washing step lowers the initial population of undesirable microbes that may compete with lactic acid bacteria during fermentation. Cut the Napa cabbage into quarters, then slice crosswise into bite-sized pieces. Next, let's prepare the brining solution. In a bowl, dissolve coarse sea salt and water. Mix it well until it is fully dissolved. Brining causes osmosis, drawing water out of the cabbage cells. This reduces water activity and softens the cabbage texture. The salty environment creates a selective condition that inhibits spoilage and pathogenic bacteria while favoring salt-tolerant lactic acid bacteria. Then, pour the brine solution over the cabbage that we washed earlier. Gently toss the cabbage until evenly coated. After that, the cabbage is wrapped with plastic wrap and weighed down with something heavy to keep it submerged in brine. This will create anaerobic conditions that favor lactic acid bacteria and inhibit spoilage microorganisms. Leave it for 1 hour and a half. Next, the preparation of starchy paste, which is the fermentation substrate. A starchy paste is prepared by cooking glutinous rice flour with water until thickened and then allowed to cool. This paste provides complex carbohydrates, which LAB can slowly break down into fermentable sugars, supporting gradual lactic acid production without added sugar. Next, gochujang is added not only for flavor and color, but also because it is a fermented ingredient that supports LAB growth. Its salt and chili compounds help create a selective environment where beneficial microbes thrive while harmful bacteria are inhibited. Moving on to preparation of kimchi paste base. Apple and onion are blended together to form a smooth paste. Apple and onion naturally contain simple sugars, which serve as an initial energy source of LAB. Using fruit instead of refined sugar supports fermentation while maintaining a more natural formulation. Ginger and garlic are added later to preserve texture and avoid excessive antimicrobial effects. Compounds like allicin and gingerol can inhibit LAB if added too early, so adding them later ensures proper fermentation. Meanwhile, fish sauce is to enhance flavor. After that, pour the blended ingredients into the starchy paste and mix well to combine. Next is secondary washing. After brining, the cabbage is rinsed 2 to 3 times with clean water to remove excess salt and then drain for 15 to 30 minutes. Controlled salt concentration is important because excessive salt can inhibit LAB growth, while insufficient salt may allow spoilage organism to grow. Now, coat the washed cabbage evenly with the prepared paste. After the cabbage is well coated with the paste, place it into a container and wrap it tightly to create an anaerobic environment that favors lactic acid bacteria. After that step is done, allow the kimchi to ferment at room temperature for 1 to 2 days, then transfer to refrigeration for slow fermentation. LAB such as lactobacillus and leuconostoc ferment sugars derived from starch and fruit into lactic acid, lowering the pH from approximately 6.5 to below 4.5. This acidification preserves the kimchi and contributes to its characteristic sour flavor.
On the second day of fermentation at room temperature, small bubbles were formed. This shows a clear result of production of gas, mainly carbon dioxide. The kimchi also shows textural changes where vegetables become softer but remain crispy due to proper fermentation. Additionally, the liquid becomes slightly cloudy. This reflects microbial activity and dissolved compounds released during fermentation. The kimchi fermentation is conducted through sensory evaluation and measurable indicators. A successful fermentation produces a pleasant sour aroma in taste, balanced with spicy and savory flavors without any sign of spoilage such as mold growth or foul odor. The color remains vibrant red, not dull or brown, and this indicates good quality fermentation. The dominance of lactic acid bacteria such as lactobacillus and leuconostoc species confirms that fermentation is functioning correctly. This bacteria lowers the pH and creates conditions unfavorable for pathogens.
In conclusion, the most direct proof is the formation of carbon dioxide bubbles that occurs during the incubation period. This is a visual signature of active lactic acid bacteria. Our kimchi fermentation experiment was a clear success. We have successfully initiated the fermentation process.